Thank you for giving me the opportunity to share my data with you. And thank you also for tuning in. Today we'll talk about how leukocyte breach the vessel wall. This process, known as transendothélial migration, is involved in several conditions in the human body. For instance, in inflammation, normal daily immune surveillance, but also cancer metastasis, and for instance, immune cell therapy. And the list can go on and on. But it's our goal in our lab to understand the molecular details of the leukocyte transendothélial migration process. Now, this is a typical text scheme that you find in the textbooks, I have to say, where the leukocyte starts rolling and adhering on the inflamed endothelial cells that line the vessel wall. And from the adhesion phase, they go towards the crawling phase, and finally they transmigrate either between two endothelial cells, which is called the paracellular pathway, or they can also do this by going right through the endothelial cell body, and then we call it the transcellular pathway. It was already Eugene Butcher in 1991 who suggested these three types of specificity and diversity, three or even more steps to this process. And this was very accurately described in this mini review in Cell, and it's still worthwhile looking at that actually. And in our lab, we use this in vitro approach to study this process in more detail. We have a flow chamber that allow us to perfuse over fluids and also introduce the cells of our liking. So there can be immune cells, neutrophils, T cells, but also cancer cells. And we can then record this with the microscope and see the differences that happens actually under flow conditions. So this is an example of that. We have a endothelial cell monolayer, and we'll see here leukocytes crawling over and crossing the endothelial barrier. You can see that the focal plane changes to a more dim phenotype, and then the leukocyte is underneath. Now, when we looked at many of these events, it turned to our attention that these leukocytes were almost searching for a perfect exit site. And that actually made us wonder, is there actually a preferred exit point for the leukocytes to cross the endothelial monolayer? And if so, what would identify such endothelial exit points? And we had the opportunity to use the lattice light sheet microscope at the Janelia Research Institute in America. And this microscope allows us to image in a very fast modus in 3 dimensions in time with very low toxicity. And when we did so, we looked at the endothelial cell morphology when the endothelial cells were transfected with LiveAct, giving us the opportunity to look at the actin cytoskeleton in more detail. And we found these amazing structures to be present and dynamically being induced in the endothelial cells upon inflammation. That is important to say. And not only that, we also found that these structures were very dominantly present at sites where leukocytes cross the endothelial monolayer. This is a volume rendered recording with the lattice light sheet of these leukocyte transendothélial migration processes. Now, looking at a bit more detail to these protrusions, we found that these protrusions are typically induced at the luminal side into the lumen of the vessel, so to say. So they were protruding dorsally from the endothelial monolayer. And in a closer look showed that these protrusions were present at junction regions. However, Phe cadherin, one of the major endothelial junction markers, is not present at these protrusions, whereas PCAM, another typical endothelial junction marker, is. As you can see here, it covers almost the whole protrusion. We quantified this, and we could see this here. Significant increase in PCAM expression at these protrusions. So we started calling these protrusions junction membrane protrusions. What about the exact dynamics of these protrusions? Now we looked in more detail to this, and we could, again, using the lattice light sheet, show this really the volume of these protrusions. And with the help of some people from Geneva, we were able to quantify the volume of these protrusions, but also the area, for instance, where these protrusions actually act at the junction region. And we found that upon TNF, but also other inflammatory stimuli, more than a doubling of the volume, so to say, the dynamics of these junction membrane protrusions. So what determines junction membrane protrusions then? Well, we looked at the different origin of the endothelium, and we found that under all these endothelial cells, so derived from umbilical cord, lung, pancreas, or the kidney, basically the junction dynamics is similar to all these endothelial cell types. Flow, however, seems to induce the dynamics of these junction membrane protrusions after 5 to 20 minutes of flow. After that, it actually decreases again back to basal levels. So do junction membrane protrusions then facilitate transendothelial migration? Again, we looked at many of these movies, and this neutrophil you can already see preferred to cross the endothelial monolayer at these sites where typically these membrane protrusions were at. And indeed, about 75 to 80% of all neutrophils preferred to cross the endothelial cells at junction membrane protrusions. A clear preference for these neutrophils for these sites. So indeed, are these JMPs preferred as exit point? Well, they are full with actin, that we know. So can we use this, this idea to induce junction membrane protrusion to look in more detail how this actually works? One of the big actin regulators are small GTPases. So we focused a bit more on how small GTPases would regulate these JMPs. One of the main GTPases that control membrane protrusions and filopodia are the small GTPases CDC42 and Rac1. And to see whether these GTPases are actively involved in leukocyte transendothelial migration, we use biosensors, and these biosensors allow us to look at the activity of these GTPases in real time. So this is based on FRET, fluorescent resonant energy transfer. And when we have high FRET, we have a high activity of CDC42, and the color will be warm. Red to even white is the highest activity of the sensor, being a high FRET ratio. Whereas blue shows us a low FRET ratio, so a low activity of the CDC sensor. So when we did this, we had endothelial cells transfected with the biosensor in a monolayer, as you can see here. This is the sensor. Neutrophils were perfused over, and then we looked at the activity of the FRET sensor. And somewhat to our surprise, we didn't see any change in the FRET ratios when neutrophils were crawling over, under, or at the transfected cells. We also did this for the Rac1, and we couldn't see much. So let me allow to explain you how we did this in detail. So here the orange dots are basically the areas where we measured the FRET ratios that were exactly underneath the neutrophil. And the blue ones were actually the FRET ratios of the whole cell. And you could see that there's actually hardly any difference appearing when a neutrophil is crawling over the particular endothelial cell, indicating that the before, during, and after the crawling of the neutrophil, there was no increase in CDC42 activity. And actually we found the same for the Rac1. There was no increase or decrease of Rac1 induced by the neutrophil that is crawling on the endothelial cell. So the neutrophils do not directly activate CDC42 or Rac1 during the crawling phase. However, what we did notice is that neutrophils preferred sites of high Rac activity to cross the endothelium. In fact, 80% of the neutrophils preferred such sites. Where the FRET ratio was high, neutrophils would cross. But the FRET ratio was already high before the neutrophil arrived there. So it was not induced by the neutrophil, the activity was already there. So if this is true, can we then drive leukocytes across the endothelium by artificially promoting Rac activity? Again, having high Rac activity before the neutrophil arrives. So we thought about a method to do that, and we came up with the photo-convertible Rac probe. This probe is linked to the love domain. The love domain is light sensitive for its activation and is isolated from plants, and it's the reason why plants actually grow to the light. So we have a Rac protein that is coupled to the love domain, and normally this would keep the Rac protein inactive. And when you then activate with the proper wavelength, the love domain opens up and the Rac protein is released and thereby exerts its action to the membrane, for instance. So we did exactly that. We transfected these constructs in the endothelial cells, as you can see here, and then illuminate a specific region of the cell with the proper wavelength. And indeed, you could see that it induces the membrane protrusions in these areas. And now we allow leukocytes to cross these endothelial cells that were illuminated with the proper wavelengths. And first of all, we can see that if we do that, indeed, we induce the number of JMPs per endothelial cell. It doubles the number of the JMPs. And indeed, also the number of neutrophils is significantly increased when Rac is activated. So we induce more JMPs and we induce more neutrophil transendothelial migration. Now, that was not all. We wanted to not only over-activate Rac1, but we also want to use endogenous Rac1 by activating one of the proteins that is actually responsible for Rac activation. So we started overexpressing an exchange factor that we know specifically localized at cell site junction. And we did so for TM1. So TM1, here as you can see here in yellow, specifically localizes at endothelial junctions at these regions. And indeed, it activates endogenous Rac1, as you can see here, with a classical Western blotting pull down. And then we can see here that with this volume rendering image that the cell that expresses TM1 has a massive actin-induced protrusion here at the junction region. The junction here can be seen here in white. And then this is depicted here as a more schematic. You have to see it like this. This is the TM1. You have active Rac here. You have a protrusion at the junction region, and the non-transfected cell doesn't show that actually. When we did so, indeed, the neutrophils were very happy by crossing the cells that transfected TM1. As you can see, you can play it again. It's very fast moving. So we quantified this, and we could see a doubling of the number of neutrophils that crossed these TM cells that show activated Rac. But we did see something else that was actually more striking. And allow me to tell you this in a bit more detail. So what we noticed is that when neutrophil crossed an endothelial monolayer with two endothelial cells that were not transfected, let's say about 20% of the neutrophils preferred such junction. However, when both cells were activated or were transfected with TM1, so both cells showed an activation of Rac1, again, only about 20% of the neutrophils preferred such endothelial junctions to cross. However, when we now had a cell that only one cell, we had a junction where one cell only had increased Rac activity and the other cell doesn't, so we have a sort of an asymmetric junction, you might say, then we see a more than 60% increase of the neutrophils that preferred such junctions, as you can see here. So if this is true, can we then push leukocyte diapedesis by inducing asymmetric JMPs, or let's say asymmetric junctions, where one endothelial cell expresses a JMP and the other cell does not? We did so by trying to overexpress active Rac constructs with just an FP in a mosaic fashion. You get something like this. You have a green cell that shows this transfected as active Rac, and the red cell that shows something else, in this case live Rac, but can be any other FP. And it results in a typical junction membrane protrusion at one side and no protrusion at the other side. So having here an asymmetric junction, as you can see. And indeed, we could see that the preference of the neutrophils was that they moved underneath the cell that showed the large protrusion. And in that direction, in both cases, whether we had photoactivatable Rac or Rac-Q61, in both cases, we had about 80% neutrophils that crossed the endothelial monolayer, the junction, in that direction. Making it this movement that neutrophils make to cross the endothelial monolayer. Now that brings me to my sort of conclusion scheme for so far, that we have such a junction that is preferred for neutrophils to cross. And these asymmetric junctions might act as exit points for leukocytes. And this is interesting because such a new asymmetric junction might be a new target site for therapeutic interventions. We already have some evidence that we have chemokines, specific chemokines, or even chemokine receptors and adhesion molecules being more present at these membrane protrusions than at the other side, allowing the neutrophil to recognize such protrusions and to actually cross the endothelial monolayer right there. Now, the last couple minutes I'd like to use to show you about the vessel on the chip technology that we currently use in the lab. These are very small chips. This is about 2 millimeters only. We can fill these areas with matrix proteins, and then here the area in between, the tube, we can fill that with endothelial cells, put it on a rotor in the overhead bin, and then we have a nice monolayer of endothelial cells that allows us to also study, for instance, permeability. So this is one of those tubes. It has a full lumen, and you can do nice endothelial stainings with it. And we have fecal here and here in white. In red we have actin, and in blue you see the nucleus. And this is basically what the neutrophil should feel like when it transmigrates, I guess. It goes right through the endothelial cell body into the tissue and can continue to transmigrate, which is one of the great things about these chips. So we started to use these chips, and we treated them with the endothelial cells with TNF. And one of the things that we typically saw immediately is here you have nice endothelial cells with almost empty inside for instance, actin. However, if it's TNF treated, then you see that the actin cytoskeleton is much more prominent. And the other thing that we notice is that the vessels actually show a smaller diameter. So the endothelial cells contract. But there are no smooth muscle cells or what other cells around. So this is really true for the endothelial cells themselves. And it's not the number of endothelial cells that actually matter. It's really the contraction that makes the diameter smaller. Well, when treating the vessels with TNF, we can show that the neutrophils started to transmigrate indeed out of the vessel wall. And even they can respond to chemokines. If we add one chemokine to one outlet and not to the other, they really respond to the site where the chemokine is. So we can induce leukocyte transendothelial migration in these chips. We also did this for T cell, and it works quite well, I have to say. And when we use a software program like Imaris, we can actually start to quantify a lot of things. We can quantify the number of cells in the cell, in the vessel, or halfway, the mid-diapedesis, or the cells that actually entered the tissue. Then we can also discriminate the distance that they actually migrate into the tissue. And we can record this in exact directions in time, so we can see a neutrophil leaving the vessel wall and actually making its way into the tissue, allowing us not only to determine the distance from the vessel, but the actual distance the neutrophil has actually traveled. And we have seen neutrophils going back to the vessel as well. This is something completely new that we could not have been investigating before with in vitro settings. So this allows us to measure even more parameters of the leukocyte transendothelial migration process. Having said that, I'd like to end again with this scheme, and hopefully I've showed you that the endothelial cells can act as a specific exit point for leukocytes by inducing such asymmetric junctions where one endothelial cell shows a protrusion that potentially expresses several trafficking proteins such as adhesion molecules and chemokines. Now, one of the most important slides, I would say, this is the thank you slides, where the people like Ivar and Lilian for the first time realized there might be an asymmetric junctions being responsible for exit points. Janine and Eike did the photo-convertible experiments and the TM experiments, and Lanette and Bram are responsible for the chip experiments that I showed you. And the other people here for the great collaborations, Martijn, Joachim, Mark Winter, David Beebe, but also the people from Genelia, making this work really something great to do. And of course, last but not least, and I think I'm still in time, there's the summer school of the EVBO, ESM, the 13th and 15th of July, and it's almost free. You only have to be a member of the EVBO, only $30, and then you're a member for the whole year, and you can join this program. So please have a look at it. It's a digital one, And I think it's a great program and you can learn a lot from it. Anyway, thank you very much for your attention, and I'm more than happy to answer any question that you may have. Thank you.